Drug discovery always begins with one foundational decision, choosing the right biological target. If that decision is weak, everything that follows becomes expensive validation of a fragile hypothesis. Years of medicinal chemistry, preclinical studies, and clinical development cannot compensate for starting with the wrong biology. For decades, target discovery relied on literature synthesis, incremental experimentation, and expert intuition. These methods remain important, but the scale of biological data has changed dramatically. Today, genomics, proteomics, transcriptomics, metabolomics, epigenomics, and real-world phenotypic data generate extremely high dimensional datasets. Each patient represents thousands of interacting biological variables. Disease is not driven by isolated genes. It emerges from dynamic interconnected biological networks. The strategic opportunity for leadership is clear. We now possess more biological data than ever before. The constraint is no longer data availability. The constraint is extracting meaningful signal from overwhelming complexity. Machine learning is designed precisely for this environment. Genomic data sets contain tens of thousands of genes. Proteomic data maps intricate protein interactions. Transcriptomic data captures dynamic gene expression across disease stages. Traditional statistical methods struggle when dimensionality exceeds interpretive capacity. Machine learning models identify patterns across these layers simultaneously. Supervised learning models trained on labeled datasets can distinguish diseased from healthy states by identifying combinations of features rather than single markers. Instead of isolating one gene, ML systems uncover composite molecular signatures. Unsupervised learning methods cluster patients into biologically meaningful subgroups based on multi-omics similarity. Diseases previously treated as uniform conditions often reveal distinct molecular subtypes with different therapeutic implications. This transforms target discovery from broad generalization into precision hypothesis generation. Deep learning further expands this capability. Neural networks, including graph neural networks, can model biological systems as interconnected networks rather than isolated variables. Proteins interact through pathways. Signaling cascades propagate across cellular environments. Deep architectures capture these non-linear relationships mathematically. Network biology becomes computationally actionable. A knowledge graph integrates published research, clinical trial, outcomes, pathway, databases, protein interaction maps, and real-world evidence into a connected structure. Genes, proteins, diseases, and drugs become nodes. Relationships between them become edges. Machine learning applied to this graph structure enables link prediction. It identifies potential relationships between targets and diseases that have not yet been experimentally confirmed. This creates strategic leverage. Instead of relying solely on manual literature review, organizations can computationally evaluate millions of potential biological associations. Models assign probabilistic confidence scores to novel hypotheses. Hypothesis generation accelerates and prioritization becomes structured. Target discovery evolves from intuition-dominated exploration into probability-informed decision-making. However, biological data is inherently noisy. High-dimensional data sets create a well-known risk overfitting. Overfitting occurs when a model learns noise rather than signal. A model may perform impressively on training data, but fail when applied to new independent data sets.
For life sciences leadership, this is not an academic concern, it is capital protection.
If predictive models identify targets based on spurious correlations, downstream investment flows into biologically weak programs masked by statistical artifacts. Preventing this requires disciplined validation, cross-validation, independent test sets, external cohort replication, and biological plausibility checks must be standard practice. Machine learning outputs must be interpreted alongside domain scientists. AI augments biological reasoning, it does not replace it. Strategic advantage emerges when computational scientists and experimental researchers operate as an integrated system. When teams work in silos, models lack biological grounding and experiments ignore computational insight. When integrated, iterative refinement strengthens both model robustness and experimental precision. Multi-omics integration becomes particularly powerful when connected to clinical endpoints. Linking genomic signatures to patient response data enables identification of targets associated not only with disease presence but with therapeutic sensitivity. This reframes discovery around downstream probability of success. Machine learning can also analyze longitudinal transcriptomic changes, capturing disease progression over time rather than static snapshots. Temporal modeling improves understanding of when intervention is most likely to succeed. Uncertainty is compressed earlier in the pipeline. Strategically, multi-omics ML integration should function as an early-stage filter. The objective is not perfect certainty. The objective is improved probability before expensive laboratory validation begins. Every weak target eliminated computationally preserves years of capital and effort. Knowledge graphs strengthen this filtering process by incorporating historical failure data. If prior molecules targeting a pathway fail due to toxicity or inefficacy, those outcomes inform predictive modeling. This reduces repetition of historical mistakes. From a portfolio perspective, ML-enabled target discovery enhances option quality. Rather than increasing the number of targets pursued, organizations improve the probability-weighted value of each selected target. Exploration does not disappear. It becomes structured. Rare and complex diseases represent another opportunity. Traditional discovery in rare indications is constrained by small sample sizes and fragmented datasets. Machine learning can integrate sparse data across institutions, identifying molecular commonalities that individual datasets cannot reveal. Federated learning allows collaborative modeling without sharing raw data, preserving privacy, while expanding analytical scale.
For CROs and biotech collaborators, demonstrating ML-driven target prioritization strengthens strategic positioning. Sponsors increasingly expect computational rigor early in the pipeline.
For large pharma organizations, internal capability in multi-omics integration reduces dependency on externally sourced hypotheses. Governance remains essential. Models must provide interpretability, feature importance analysis, pathway mapping, and biological validation. Experiments must accompany computational outputs. Blind trust in algorithmic ranking is as risky as ignoring it. Leadership posture should be confident but disciplined. Machine learning does not eliminate biological uncertainty. It reshapes how uncertainty is quantified. Investment must extend beyond algorithms to data quality, high-quality curated data sets. Standardized pre-processing pipelines and consistent metadata structures determine model reliability. Data governance becomes discovery infrastructure. Biological complexity continues to increase. Data volume continues to expand. Human cognitive capacity remains constant. Machine learning extends analytical capacity beyond human limitation. When applied rigorously, ML converts overwhelming biological noise into structured probability. The organizations that lead in the next decade will not be those with the most data. They will be those with the strongest signal extraction discipline. Target discovery is the foundation of the pipeline. Improving precision at this stage amplifies value across every subsequent phase. Reducing false positives decreases preclinical attrition. Enhancing biological relevance improves phase 2. Responder rates strengthening mechanistic clarity improves regulatory confidence. These effects compound. Machine learning in multi-omics integration is not an optional research experiment. It is strategic infrastructure. When implemented with validation discipline, scientific collaboration, and governance maturity, it transforms discovery from exploratory art into probability-engineered science. In an industry where each development program represents significant capital and years of patient expectation, improving signal extraction at the beginning is one of the most powerful interventions. Leadership can make the data exists, the algorithms exist. The strategic decision is whether to integrate them deliberately to redesign the foundation of the pipeline. When the quality of the first decision improves, every decision that follows becomes stronger. That is how machine learning shifts target discovery from uncertainty management to probability engineering.